Now we are going to take into account DNA replication. When we had talked about the central dogma of molecular biology, over there we discussed that DNA replication takes place. DNA produces its copies by the process of replication. As you know, it was proposed by Crick. Apart from that, you have an idea that during the S phase, the entire DNA content is replicated and new DNA is formed for the newly formed cells. So, this is what is going to be our topic of understanding in this lesson. But over here, we are not going to get into details of the process, but we are going to see how it was proven that DNA replication is semi-conservative. Now, we come across a term known as semi-conservative. What is conservative? Conservative is something that likes what is old. And what does semi-conservative means? That half of it should be old, half old thing would be liked, and half of it would be new. When Watson and Crick were proposing the double helical structure of DNA, this thing came into their mind that there lies complementarity in the structure of DNA. So they had it in their mind that somehow they are paving a way for the replication process as well in the structure itself. So when they said it, they also told that DNA would be replicating itself and the replication would be in such a manner that the old strands would form the new strands. From the old information, the new information would be taken up, and as the name suggests, it would be semi-conservative. I am just giving you an idea. Supposedly this is your DNA, alright. This is old strand, this is also old strand. The new DNA that would be formed out of it will be in such a way that a new complementary strand for the old strand is synthesized. This is just an idea, we get into details. So supposedly this has to give rise to two DNAs. The two DNAs which would be formed will be like this. The new complementary strand would be with the old one. The newly formed will be formed on the basis of complementarity with the old one, and similarly the next one will be like this. So you see that this was the original one. Those which have been formed newly, they are containing half of the content that is old and half of the content that is new, and hence this term semi-conservative. Alright, now how it was proven? In 1958, there was an experimental proof given that DNA replication is conservative. Now, as I told you, it is like this. Now, this has to be proven. Again, E. coli comes to our rescue. E. coli has a life period of 20 minutes. You know that it divides within 20 minutes. So such a rapid rate of division could be of help to the scientists. That's why they choose E. coli oftenly, and moreover a lot of studies have been done on E. coli. So it becomes an important experimental aspect in this molecular biology. E. coli experiment for proving that DNA replication is semi-conservative was carried out by meselon and stahl. Okay. Alright, these are the second names of the two scientists. In 1958, they carried out this experiment using E. coli and the idea of density centrifugation. See, when you sit for exams, this is important topic. So you need to remember the things. I am just explaining this from the examination point of view, keeping in mind that I am a teacher and I have to enable you get good marks. Apart from that, from understanding point of view also, keep in mind that this experiment is quite crucial. Okay. Now, this density centrifugation was carried out using cesium chloride. What is density centrifugation? That in a test tube you will give the medium, that would be cesium chloride, and whatever you want to see or whatever you have added, they would float or show up in the medium according to the density. So, you are going to get gradations in the column, okay, according to the density. That is why it is known as density gradient centrifugation. You get a gradient according to the density of the substances which are present in cesium chloride. Now, what is done in the experiment is that E. coli is first of all grown up for considerably 1 day on a medium that contains radioactive ammonium chloride, okay. So, our aim is to grow E. coli on radioactive nitrogen, heavy nitrogen, okay. It is grown on NH4Cl14, right. This this is the medium on which it is grown, and it contains radioactive nitrogen, which makes the DNA of E. coli radioactive, okay. First of all, it is grown for a long time. All the E. coli which have grown on this, at least the later generation, they will have radioactive DNA. Why? Because DNA uses the nitrogen. Now, when there is radioactive DNA, enough of E. coli has been obtained which contains radioactive DNA. What would be done is, then the E. coli would be grown on simple medium which do not contain radioactive nitrogen. So, the new DNA strands that would be formed, or those would be formed, they will not contain radioactive substance. So, this was proven how? First of all, they took the first E. coli, first batch of E. coli which was grown on radioactive material, and they centrifuged it in cesium chloride. And what they got, supposedly this is your test tube, okay. So, the E. coli that they got in the density gradation was, because it was heavy isotope of nitrogen being used, we had a band of heavy DNA, okay, a band of heavy E. coli that stood downward in the gradient, and we get this because all the E. coli had radioactive DNA. Then what was done is, after 20 minutes had passed, growing the E. coli on that medium which did not contain radioactive nitrogen, what we saw was there was formation of a hybrid band which lied between, somewhere between heavy and light. This is what they got, not light but somewhere between heavy and light. So, this was the hybrid band, hybrid DNA. After 20 minutes later on, when further division took place, the E. coli are growing on normal nitrogen, ammonium chloride containing medium. Now, the hybrid stays over there and you get a light band as well. This was the hybrid. Now, you are going to get a light band. Hybrid is there and light DNA is also coming. After 20 minutes, now as you keep on observing and centrifuging, you are going to find hybrid always there, and the width of light band is going to increase more and more after 20 minutes, okay. It is going to increase further 20 minutes, it is going to increase further. So, this is what was observed in their experiment using E. coli. Now, supposedly initially this was the radioactive DNA, alright. As a result of semi-conservative replication, hybrid is formed. So, one strand stays like this, while the new one is non-radioactive. Previous one stays like this, while the new one is non-radioactive, and hence the hybrid. Now, this DNA is going to give rise to more strands. and what it would be like, the hybrid is going to stay over there. This red is going to give rise to a new blue. Then we have the hybrid, this one, it is giving rise to a new, and then we have this blue. This would also act as a template. Again we have blue, this would also, and we have this. So we get light as well as hybrid. What would be the next stage? The previous one would stay, of course. This would form, stay, and a new would be formed. Then again this blue like this. Then we have the red again. From this a new blue would be formed. Then we have that blue. From this new blue, we have these blues, new strands, and blues continue forming new strands further and further and further. So this experiment proved that somehow the previous strand acts as the template for formation of new strand. Okay, and hence the DNA replication is semi-conservative. It was proven by this experiment, and you can very well make it out that if we are given the example of E. coli, what happens to the DNA content? What would be the ratio of non-radioactive and radioactive content? Over here you get 1:1, that is the ratio, alright. 50% of them are new non-radioactive and 50% are radioactive. Out of these you get the ratio of 1, 2, 3, 4, 5, 6, 6:2, that is 3:1. Alright, after this 4 are going to give rise to 16, so you are going to get 14:2, that is 7:1. Then you move further, it goes on increasing, but 2 do stay there. That means the replication is semi-conservative. That is why we don't get rid of the hybrid band even after many successive divisions, okay, which are carried out in 20 minutes. So this was all about the experiment carried out by meselon and stahl to prove that the DNA replication is semi-conservative.